Welcome to this technical summary on DNA framework nanoreactors for programmable membrane fusion and their applications. This presentation will cover design, structural studies, dynamic fusion analysis, single vesicle quantification, and cell delivery experiments from a recent scientific paper. The question addressed by this paper is how to create a programmable, modular system to study and control membrane fusion events at high structural resolution and with tunable parameters. Membrane fusion is a fundamental biological process critical in neurotransmission, intracellular trafficking, and drug delivery, yet the transient intermediates and energetics remain challenging to capture and manipulate precisely. The authors designed a DNA-based nanoreactor platform using DNA origami frameworks shaped as soccer balls, called DNA soccer ball frameworks, or DSFs, that serve as scaffoldings to arrange lipid vesicles in defined spatial configurations. Key parameters such as window geometry, intervesicle distance, and protein incorporation can be programmed, enabling controlled vesicle docking and snare protein-mediated fusion within confined environments. This approach allows direct structural capture of fusion intermediates via cryo-electron microscopy and supports modular assembly for higher-order fusion products. Moreover, this platform serves as a versatile system for mechanistic membrane studies and holds promise for targeted cargo delivery in therapeutics, such as siRNA transfer. In summary, this study proposes a novel DNA framework-enabled vesicle system, combining precise structural control with functional membrane fusion capabilities, bridging structural biology and synthetic membrane engineering. Figure 1 presents an overview of the DNA nanoreactor platform design and its assembly. Part A illustrates the programmable platform based on a DNA soccer ball framework, or DSF, which exhibits a truncated icosahedron geometry comprising 12 pentagonal and 20 hexagonal windows. This isotropic cage provides docking portals that confine uniform vesicle formation and enable window-mediated membrane fusion. The modular design allows tuning of key parameters such as docking strand sequences and interframe distances. Part B shows atomic force microscopy images of DSF monomers and dimers formed via close docking at hexagonal windows, confirming high-quality assembly. In Part C, negatively stained transmission electron microscopy images illustrate the DNA framework vesicles, or DFVs, encapsulating uniform liposomes in monomeric and dimeric states without snare proteins. These images verify that vesicles are well confined within the DNA framework. Lastly, Part D demonstrates the high dimerization efficiency of DFV-AB via SDS agarose gel electrophoresis, with about 85% yield of dimer formation. This is achieved through hybridization of single-stranded DNA handles extended from window edges, which provide programmable portals for vesicle docking. Overall, Figure 1 establishes the robust construction and controllability of the DNA nanoreactor system, laying the foundation for subsequent studies of membrane fusion within this confined, modular architecture. Figure 2 focuses on the structural characterization and statistical distribution of membrane fusion intermediates mediated by canonical snare proteins within the DNA framework vesicle nanoreactors. Panel A schematically illustrates the setup where DFV-AB dimers each contain snare proteins embedded in the lipid membranes. VAMP2 included in the DFVA vesicles and T-snares, syntaxin 1, SNAP25 complex, in the DFVB vesicles. These proteins cooperatively zipper to drive fusion, tracked here also by the lipophilic R18 dye. The authors successfully reconstituted snares during detergent-mediated lipid templating and confirmed the uniform structure and protein incorporation by electron microscopy and silver staining, with about 33 VAMP2 copies per DFVA and 19 T snares per DFVB. Panel B presents representative cryo-EM images of DFVA-B dimers after 18 hours incubation, revealing 6 distinct fusion stages, ranging from initial vesicle tethering to complete fusion. These were defined quantitatively using 2 geometric descriptors: DIN, the shortest distance between inner leaflets, and DOUT, the lateral separation between outer leaflets after fusion. Histograms of these parameters map 6 morphologies, permitting nanometer-scale resolution of fusion intermediates. As a control, DFVA-B dimers without snares showed mostly tethered but unfused vesicles, underlining the essential role of snare complexes in overcoming fusion energy barriers. This confirms the nanoreactor platform's ability to capture and quantitatively analyze fusion intermediates, and emphasizes the biological relevance of snare-driven membrane fusion within the DNA framework environment. Figure 3 investigates how geometric parameters of the DNA framework influence membrane fusion kinetics and efficiency. Different configurations of interframe distance and window geometry were studied, with corresponding R18 fluorescence traces quantifying fusion activity. The authors compared 3 systems: DFVH-CAB with close linker spacing, 2 nanometers, DFVH-FAB with far linker spacing, nearly 7 nanometers, and DFVN-LAB lacking interframe linkers, allowing random vesicle encounters. Real-time fluorescence monitoring revealed all showed initial fusion-associated fluorescence increases within 30 minutes. However, DFVH-FAB exhibited rapid attenuation and signal growth after that, resulting in lower final fusion levels. The no-linker sample showed minimal fusion, similar to a snare-deficient control. Comparison of half-time productivity indices demonstrated that closer tethering promotes higher fusion efficiency and kinetics. Endpoint analysis quantified the rounds of fusion, ROF, showing about 27% fusion in the close linker system versus 11% and 8% for far linker and no linker groups respectively, highlighting how increased spacing hampers fusion probability. Additionally, multivalent DFV assemblies with different stoichiometries were imaged by NS-TEM and cryo-EM, showing fusion incrementally increases vesicle diameter, consistent with mathematical scaling. Bulk fluorescence demonstrated that fusion efficiency increased with B to A vesicle ratios and exceeded that of freely mixed vesicles due to spatial confinement. However, linearly assembled DFV arrays formed via opposite window docking unexpectedly showed reduced fusion efficiency, likely caused by geometric and stoichiometric effects. Altogether, Figure 3 reveals that DNA framework geometry rigorously governs membrane fusion dynamics, providing quantitative control over fusion probability, multiplicity, and kinetics by tuning spatial parameters and assembly architecture. Figure 4 presents high-throughput single-vesicle quantification of DFV-mediated membrane fusion using nanoflow cytometry, or NFCM. Panel A depicts the dual-color labeling strategy where DFV AVAMP2 vesicles are tagged with FITC cholesterol and DFV BT snare vesicles with Psi5 cholesterol, enabling direct co-detection of fusion based on fluorescence coexistence rather than intensity changes like lipid dye dequenching. Representative fluorescence scatter plots, before and after DNase digestion, are shown in Panel B. Prior to digestion, DFV nanoreactors assemble into monomers, dimers, and higher-order species with distinct FITC and Sci5 fluorescence populations. DNase treatment removes the DNA framework, resulting in disassembly into individual vesicles. Post-digestion, the double-positive FITC plus Sci5 plus population corresponds to vesicles with merged membranes containing both fluorophores, indicating true fusion events. Panel C illustrates how the proportion of fused populations increases progressively with higher B to A ratios, consistent with previous bulk fusion measurements. Importantly, multiple well-separated Sci5 intensity subpopulations emerge within the double-positive gate, reflecting vesicles that underwent multiple fusion rounds. Panel D quantifies the fractions of vesicles fused 0 to 4 times, showing strong agreement with rounds of fusion values derived from bulk kinetics. Finally, Panel E analyzes size distributions of vesicles from different fusion rounds, revealing a stepwise increase in median diameter that closely matches cryo-EM measurements, confirming the geometric accuracy of fusion intermediates and highlighting the robustness of the NFCM approach for quantitative membrane fusion studies at single particle resolution. Figure 5 explores the functional application of DNA framework vesicles in living cells, demonstrating membrane fusion and effective cytosolic siRNA delivery. Panels A and B present confocal imaging of HeLa cells expressing VAMP2, interacting with R18-labeled DFV at-snare vesicles under close tethering conditions, with and without cytochalasin D, Cyto-D, a potent endocytosis inhibitor. Under Cyto-D treatment, Panel A, strong continuous R18 fluorescence outlines the plasma membrane, indicating efficient membrane fusion without vesicle internalization. Without Cyto-D, Panel B, intracellular punctate R18 signals also appear, reflecting endocytosed vesicles undergoing fusion in endosomal compartments. This verifies membrane fusion driven by the DNA framework, occurs predominantly at the cell surface but can also proceed post-endocytosis. Panel C illustrates the siRNA incorporation strategy, where siEGFP molecules are linked to DFV at-snare vesicles via cholesterol-modified DNA linkers, enabling controlled localization on the vesicle surface or lumen. Panel D provides a mechanistic schematic and confocal images showing protection of interior siRNA from RNase A digestion, and the effect of Cyto-D inhibiting endocytosis. Flow cytometry data in panels E and F quantify EGFP gene silencing efficiency. Notably, under endocytosis-inhibited conditions, DFV at-snare vesicles achieve nearly 52% knockdown of EGFP expression, which remained similar after RNase A treatment that degraded extracellular siRNA, but not encapsulated molecules. These results strongly indicate direct cytosolic delivery of siRNA via membrane fusion rather than endocytosis-dependent uptake. This confirms the versatile utility of the DNA framework nanoreactor for targeted cargo delivery in cells through programmed membrane fusion. In conclusion, this paper introduces a programmable DNA framework-based nanoreactor platform that precisely assembles lipid vesicles into defined geometries, enabling controlled snare-mediated membrane fusion under confined conditions. The DNA soccer ball framework serves as a versatile nanoscale scaffold, supporting tuning of intervesicle distance, docking window arrangement, and multivalent assembly. High-resolution cryo-electron microscopy captured 6 distinct fusion intermediates, characterized by quantitative geometric parameters, elucidating the dynamic fusion pathway with nanoscale precision. Bulk fluorescence assays and single-vesicle nanoflow cytometry analyses quantitatively correlated rounds of fusion, kinetics, and efficiency with framework geometry and assembly stoichiometry. Notably, multivalent and linearly extended assemblies demonstrated predictive fusion behaviors, while close tethering promoted fusion over distant or random linker configurations. Finally, the nanoreactor system was successfully applied in living cells to achieve direct membrane fusion and cytosolic delivery of functional siRNA, bypassing endocytic internalization mechanisms. These advances not only deepen mechanistic understanding of membrane fusion, but also pave the way for synthetic cell engineering and targeted therapeutic delivery platforms. The DNA framework vesicle nanoreactor thus represents a robust and adaptable tool to study and harness complex membrane fusion processes at multiple scales, with broad implications in biophysics, nanotechnology, and biomedical research.